Right. Thank you, Edward. I'm Sean. I'm a post-PhD student in the Vendruscolo group at the Department of Chemistry. Firstly, many thanks to CSAR for giving me this opportunity to share a little bit about my research. Today, I'm going to talk about therapeutic approaches in protein misfolding diseases. Now, the first thing we want to clarify here is what exactly do we mean by a protein misfolding disease? And for that, we go back to proteins. Now, proteins in our body are made up of what we call nascent polypeptide chains, the stringy things that you see here. Now, they undergo a very natural phenomena in which they sort of collapse upon themselves to form a geometry or shape of a protein. And this this kind of shape and structure allows it to interact with many different partners and achieve its function. However, sometimes these proteins do not fold correctly. We call this process misfolding. Now, when they misfold, they like to clump up together and form what we call fibroaggregates. And these aggregates that you see here, these clumps, are hallmark features of many protein diseases. So the most familiar disease that we know of is Alzheimer's disease. So you see these clumps in the brains of patients with Alzheimer's disease. This also happens in Parkinson's disease, Huntington's disease, so on and so forth. But it can also be found in systemic disorders, such as in the heart, in your eye, and in your pancreas as well. Unfortunately, especially from these kinds of diseases, such as neurodegenerative diseases, there's a lot of research effort to be tried to find therapeutics against these diseases. But despite the amount of money that's being put in, the kind of outcomes is not favorable. For example, in Alzheimer's disease, there's currently no drug that is able to slow down the progression of the disease nor cure the disease itself. So what is the whole problem of this drug discovery approach against things like Alzheimer's disease? Why is the failure rate so high? And so for that, we must look at the very fundamental core of this problem. And that's how we do drug discovery for these protein misfolding diseases. Now, traditionally, if you sort of recall your high school biology, small molecules or therapeutics are used in a very lock and key kind of way. So you have the protein that you want to target, the protein that you know is the cause of the problem. So you develop a small molecule, the small molecule binds towards the proteins, and you actually activate the response or you deactivate the response of the protein. And you're well and done. This is very successful in enzymes, in signaling receptors, so on and so forth. And that's because you have only one kind of structure of this protein, and it stays the same throughout the entire time. It is static. Now, when we move into a protein misfolding problem, this becomes a lot more complicated. So first and foremost, protein misfolding is a dynamic process. So it doesn't happen. That's why things like Alzheimer's disease, for example, the occurrence of Alzheimer's disease is age-dependent. It happens to older individuals rather than younger ones, and that's because this is dynamic. The second thing is because many structures can occur over time. As you can see here, it's not a one-step process from your nascent native protein into your aggregate. It undergoes many different steps, and which finally gives you your fibroaggregate that you see here. So if we use our traditional drug discovery techniques, it's quite difficult to identify what's the problem. Furthermore, the complexity increases even more because as we find out for most of these protein misfolding diseases, this fibrillar aggregates that I've been describing earlier is a downstream process. It is something that has already occurred after the damage to your brain has already been done. As it turns out, these intermediates, or what we call oligomers, for example, are the most toxic species that can cause all the problems in the pathology of the diseases rather than the final product of the fibrillar aggregate. So like I said, now if you have a molecule that sort of targets the protein in solution, you just simply put it in. What you're going to get is you're going to get a response. But what is the response pertaining to? Is it pertaining to your monomeric substance? Is it your monomeric substance over here? Is it the final fibrillar aggregate? Or is it the intermediate structure? What you really want is a systematic kind of way to be able to design molecules and design therapeutics to be targeting your intermediates over here. So many people will ask, okay, so let's then, come up with a technique that allows us to find these intermediates, and then you can put your small molecules in or your antibodies in, and then you can solve the problem. Unfortunately, most of the techniques that we have, which are very standardized, measure solutions in what we call bulk. They give you a very discrete kind of information over time. So basically, you get this kind of signal that tells you the number of fibrillar aggregates that you have over time. But like I said before, it doesn't give you any information about the intermediates that form before that. Thankfully, at the work that we have done at the Center for Misfolding Diseases, the way to solve this is to apply physical chemistry and very complicated math. So, to save you the complicated math part, basically, what you can say is that the kind of experiments that we have by using mathematical modeling and using rate equations to describe processes, you can now identify how do intermediates form before the final fibrillar aggregates form. And so with that, by applying different kinds of differential rate laws, you can actually try to identify how does the protein aggregate over time. And so you can deconstruct this to find out at a certain period of time how much of the starting material you have, how much of the ending material, the fibrillar aggregates that you have, but more importantly, how much of the intermediates that you have. So my PhD work has mostly been extending this work in the use of small molecules in targeting these intermediate substances. So basically, the experimental approach is to put the small molecule with the protein in solution. Now, as you can see here, the aggregation over time is delayed in the presence of the molecule, which is good. It means the molecule is working. Now, what do we do next after the experimental approach is to use complicated mathematical modeling. And so you can then find what's the difference in the intermediate species being formed over time in the presence of these molecules. Then now we can use very medicinal chemical concepts to apply them into our chemical kinetic strategy. And so you can find different kinds of molecules. So as you can see here, this is quite a library of molecules which are all chemically quite similar. And you can find out how do these intermediate changes in the presence of all these molecules. In fact, from there, you can rank them. So you can say, okay, molecule A is more potent than molecule B, or molecule C is more potent than molecule D. So now you have quite a rational strategy that allows you to identify which part of the molecule is responsible for its inhibitory potency. And you can use that to apply to different kinds of derivatives, different kinds of scaffolds, and design libraries, not just a single target, not just a single molecule, but libraries of compounds that can work against aggregation. Finally, we have also validated them in vivo. So this is in worm models expressing the aggregation. So as you can see here, short AD worms as Alzheimer's disease worms. Because of the aggregation, they're paralyzed, and you can see that they're hardly moving, and most of them are dying. But in the presence of the drug, you can see that the motility is restored because the aggregation is being prevented due to the molecules. So I've been very fortunate to be involved in many kinds of studies pertaining to small molecules. So we do find that we can use them in many different techniques. One of them are in traditional chemistry techniques like fragment-based screenings, and also in our kinds of method developments towards small molecules. Furthermore, we can also use this to try to understand how does aggregation happen in the brain. Because now, as I described earlier, what we have are solutions of buffer with protein and small molecule. That's it. And naturally, we know that in the brain, it contains a lot more. It contains metal ions, it contains lipid membranes, it contains other proteins. So we want to try to work towards understanding how does aggregation happen in a physiological environment. Finally, so due to the work that we have done here, there's been a spin-off company known as Therapeutic. It's a biotechnology company whose proprietary approach is, as I've explained earlier, based on the techniques of using mathematical modeling and physical chemistry to describe the behaviors we have in biology. And hopefully this biotech company will be able to quantify and determine and build small molecule libraries to help us combat a lot of these protein misfolding diseases, such as neurodegeneration and metabolic disorders. So finally, I'd like to just acknowledge the following people who have been instrumental in my PhD work. More importantly, my supervisor, Professor Michele Vendruscolo, my day-to-day supervisor and my mentor, Dr. Johnny Hapshi. I'm also very thankful to Professor Thomas Knowles and the Lund University in Sweden, Professor Sarah Lindsay. Finally, I just wanted to dedicate this presentation to Professor Sir Christopher Dobson, who was one of the leaders of the chemistry of health building. He was a great inspiration to all of us, and he has really encouraged us to be better scientists and better people. So for that, I dedicate this presentation to him. Thank you very much.